Hello friends, today I am going to tell you about transcription in prokaryotes. Now what is transcription? Transcription is the process of synthesis of RNA from DNA using DNA as a template. So RNA is synthesized from DNA where DNA acts as a template. Now, what are the requirements of RNA transcription? The first it is, as we told, DNA will be there. I told you earlier it is a template. So, DNA is there. Second is an enzyme which is known as RNA Polymerase. And the third is the nucleotide triphosphates. So these are the three things required for the process of transcription in prokaryotes. Now let us see the enzyme first, RNA polymerase. Now, what is the structure of RNA polymerase? The structure of RNA polymerase is as follows. 1. It is made up of 6 sub-units. They are 2 alphas, beta, beta dash, omega and sigma. Now, they are arranged in this way. 1, 2, 3, 4, 5 and 6. Alpha, alpha, beta, beta dash, omega and sigma. So in this way they are arranged. So when all the 6 subunits are present, RNA polymerase is known as holoenzyme. When all the 6 subunits are present, it is known as holoenzyme, and when the sigma factor comes out, it is known as the coenzyme. So that means in coenzyme there are only 5 subunits. Now let us see the next requirement. The next is a DNA template. Now we all know that DNA is double-stranded structure. We have 2 strands of DNA, whereas RNA is a single-stranded structure. So only one of the strand of DNA will be copied, and that strand will act as a template. This is a double strand of DNA. One of the strand of the DNA will act as a template. Now, which strand will act as a template? As we all know, the nucleotides, they are produced in the direction of 5' to 3'. So, in that case, this lower strand, it will act as a template, and thus this strand will be known as the template strand or sense strand. The lower strand, which will act as a template, it will be known as a template strand or the sense strand, whereas the other strand, it will be known as nonsense strand. This will be known as the nonsense strand, or it will be known as the coding strand. Now, why this one is known as a coding strand? Let us see that. Now we find the bases present over here. Now suppose we have A, we have T, we have C, G, C, T, A, T, C, G. This is the base sequence on the lower strand, that is the template strand. Corresponding to it, we'll find the base sequence in this way: T, A, G, C, G, A, T, A, G and C. Now, RNA will be formed using this strand, the 3' and 5' strand as a template. So, this is suppose the RNA strand. It is being synthesized and copying the base, taking the base sequence from its template. So what we find, against A, it will be U, A, G, C, so what do we find? What do we find? That this sequence resembles the newly formed RNA, resembles the upper strand, and that is why this is known as the coding strand. So this strand is coding because the newly formed RNA, the base sequence of the newly formed RNA is complementary to the coding strand. Now the nucleotide triphosphate is present in the cytoplasm. The entire process of transcription completes in 3 stages. The first stage is the initiation, second is the elongation, and third is the termination. First is known as initiation. Second, it is known as elongation, and the last is known as termination. Now let us see initiation first, how it takes place. This is a double-strand DNA. Now the whole entire strand is not transcribed during mRNA transcription. Only a part of it is transcribed, and that part is known as the transcription unit. Now this is the +1 phase from where the transcription starts. Now just opposite to the plus phase is the negative strand, and that is known as upstream. And the direction in which transcription process goes on, it is known as downstream. Now for the process of initiation to begin, this part, the upstream part, is very important because here we find the promoter which promotes the process of initiation. The promoter is here. Now -10 base pair, like from here if we start -1, -2, like this. So when we come up to -10, we have the promoter which is known as the Tata box. It has the sequence TAT, AAT, and it is known as the Tata box or the Pribno box So this at 10 base pair, we find the promoter which is known as the Tata box having the sequence of base in this form, and we also know it in the form of Pribno box after the name of the discoverer. At minus 35 base pair, we find another sequence which has TT, GA, CA. This is the consensus sequence. So on the upstream, so this is the place where transcription will begin. It is the first +1. Now this is the end portion where the transcription will end, and here we have the terminator. The terminator has a inverted sequence and a sequence of A at the end. So at the end side towards the downstream where the transcription is going to be over, that is known as the terminator, because everything has to come out to an end. When RNA is being synthesized, there should be some place where the RNA synthesis has to stop, and that is being stopped by the terminator present downstream, about 100 base pair we expect. So now we'll see the process of initiation, how it begins. This is the initiation site or the promoter site, these two. So what will happen, the RNA polymerase will join over here at the promoter site and will form a closed complex. The sigma factor will also unite with it. The sigma factor, it will also join with it. This is the sigma factor, and the RNA polymerase is a complete enzyme, holoenzyme. It has got all the 5 subunits and the sigma factor. The sigma factor is able to recognize the promoter. So in the beginning it is a closed complex, but later on the hydrogen bond between the nucleotide base starts to melt, and then an open complex is formed. When the hydrogen bond between the two nucleotides, like here we have the nucleotides and the bond is there, so the bond will now break and it will come into an open complex. So let us see the open complex. Now this is an open complex where the bond between the nucleotide base has broken down. So it will be in this form. These are what? They are the bases. The polymerase is here. Now they have opened the bond and now what will happen? The synthesis of RNA will start in 5' to 3' direction. Here it will start, RNA will start forming. They will read the base and the corresponding nucleotide bases which are present in the cytoplasm. They will move over here and attach with the template base. So if it is A over here, since it is a ribose mRNA, U will come over here, T, C, G, so what will happen? The base will be T, A, G, C, C, G, T, U and U. This will be formed when the base pair is about 10 base pairs are being formed. What will happen? The sigma factor will dissociate and now the holoenzyme will be a core enzyme and the process of elongation will begin. So what will happen when on the newly synthesized RNA, if there are 10 base pairs are formed, what will happen? The sigma factor will dissociate and what will be left? The RNA polymerase will be now in the form of core enzyme. It will be a core enzyme. So in this way now the process of elongation will begin and the RNA will be formed, a chain of RNA, it will be formed. In this way it goes on forming. Now once the polymerase reaches the termination site, RNA strand is continuously being formed by the process of elongation. The new bases are being added to it and it is elongating in length. Now what will happen? RNA, here is the termination site. So RNA polymerase will now reach the termination site. This is the termination site and here the RNA is being synthesized in 5' to 3'. Now what happens at the termination site? So now the RNA has been formed and it has to end. The strand has to end. That means the adding of base pairs should end at this point. So this process is known as termination, and the process of termination can take place by two methods. The first method, process of termination can take place in two methods. The first, it is RO independent, and the second is known as RO dependent. Now RO independent is the form of termination where we do not need any enzyme from external, from outside. It is an intrinsic termination. This is also known as intrinsic termination because we do not need any enzyme from outside. So now let us see how it happens. This is a DNA structure. Here the base pairs are there. Now this is the normal base pair. This one is a CG rich base pair, this area, and it is known as inverted pairing. Now see, in between we can have GTA, CT, CA, T, GA. Over here we'll have G, C, G, G, C, C, G, G, C, C. Here we have G, G, C, C, G, G, C, C, G, C. So this is an inverted, this and this you can see, and these two, they have got a inverted base pairing. So what happens, the center part, it comes down. The force of attraction, the hydrogen bond is very strong with the force of attraction, and the hydrogen bond is very strong between C and G. So what will happen, this will come down in this way. It will form a hairpin loop like structure. This will be, as it is a RNA, it will be formed C. Yeah, before that, let me tell, this is a DNA sequence. So first we'll form an RNA corresponding to it. We'll form an RNA corresponding to it. Now this is the RNA. This is the RNA, a chain of RNA which has newly been synthesized. Now this chain has to be terminated. Once the RNA polymerase reaches this area, we can see that this one and this one is an inverted base pair. So what happens, this comes down in this way and forms a hairpin loop like structure. Now let us see this structure over here. I'll draw it separately. So first we'll understand what is this. Now in the center we have this base pair G, U, A, C and U. On this side we will have CGCC, CGCC, GG, CCGG. Here we find G, C, G, G, C, C, G, G and CC. Now they have got a very strong bond between them. This is the bond. So as the bond is very strong, they attract with each other with a quite force, with a jerk. What will happen after that jerk? We have this weak bond over here. It is A and U over here. The bond between A and U is weak. As the strong bond attracts with them with a force, the bond between A and U will break and the ribosome will be liberated, whereas the mRNA will also be liberated. So at the end, the RNA which will be liberated in the cytoplasm will be like this. In this method, the RNA comes down in the cytoplasm. So this is the first method of termination, which is RO independent, Rho independent, RHO. Now second type of termination is Rho dependent, where we need an external enzyme which is known as Rho protein. We need an external enzyme which is known as Rho. Now let us see what happens. Now RNA polymerase reaches the termination site. RNA is being synthesized. The hair loop structure has been formed and now the chain is here. At one place there is a long chain of C, cysteine. It is a C area which is known as RUT, Rho utilizing site. This area is known as RUT, Rho utilizing site. So at this place, the Rho will come and attach in this way. Now Rho consists the enzyme hexokinase. There is a bond between the GC rich compound, This Rho, it will start moving towards the polymerase enzyme, and it will, while on its way, it will dissolve the hydrogen bond over here, come upstairs and reach the polymerase. And once it reaches the polymerase, it dissolves, the RNA will come out and the RNA polymerase will go out. In this way, the strand of RNA will be formed. So the Rho protein, it consists of the enzyme hexokinase, which moves towards the RNA polymerase, and at the termination site, it separates the RNA polymerase from the template. RNA polymerase goes out and the new RNA strand is left in the cytoplasm. So in this way, the termination process of RNA is completed, both Rho dependent and Rho independent. So now we can see that mRNA has been transcribed. Thank you.